Can you see it?
Yeah, cool.
Okay. So I already guessed I'm Jing Wang from Energy Engineering College of Zhejiang University, China. I am very honored and happy to introduce our recent research content for you. Our research title is a novel calculation model for microalgal growth prediction in any photobioreactors based on photosynthetic electron transfer. And I completed research with my tutor, Professor Jun Cheng and other four lab mates, Danny Guo, Le Chongchen, Xuan Xiangyu, and Yang Tang. First, I will give you some information about the background of our research. With the increasing attention to carbon neutrality, microalgae with high photosynthetic efficiency and growth rate have become the ideal material for biological fixation of carbon dioxide. But no matter in laboratory or in the industry, we often find some problems that the current theory cannot explain perfectly. There are too many types of photobioreactors. They all have different maximum biomass yield with the same microalgal strain. Even for the same microalgal strain, when they were cultivated in two different shaped bottles, they will have different growth rate and maximum biomass yield. Therefore, data from different experiments could not be compared, and the relationship between them was also unclear, affecting the researchers' judgment on the experimental results of microalgae from numerous literatures, which brought a great bottleneck to the study of microalgal carbon fixation. Here, we established a calculation model to explain this phenomena. It can be used to predict the photosynthetic growth of microalgae in any reaction system based on photosynthetic fluorescence theory. So let's start from photosynthesis. In the case of photosynthetic autotrophic, the growth and biomass accumulation of microalgae are fundamentally determined by the intensity of photosynthesis, and the electron transfer efficiency of light reaction determines the number of electrons available for the dark reaction, thus deciding the carbon fixation efficiency of the dark reaction. So there is a classic indicator proposed by previous studies to show the actual photosynthetic capacity of light reaction in a certain instant. That is ETR, the effective photosynthetic electron transport rate. It has been widely used in thousands of studies related to plants and microalgae. And here is the formula of ETR, composed of a light intensity. In this formula, it was written by PFD and so-called 5 PS2 and two constant. One is F1. The value of F1 is always 0.5, and another constant named F2. We mostly concerned about F2 because in this formula, it must represent the light energy absorption proportion of plant or microalgae in a certain area. In its formula, F2 was usually replaced by the constant of 0.84. However, these constant value always lacked practical and effective measurements, especially in microalgae research, because microalgae grow in the water, so the light absorption properties are always changing with the cell proliferation. A static number would be far from the actual situation, but so in our research, we provide a new calculated formula for estimating F2 of microalgae. It was firstly created from the principle of optics is here. all parameters in this formula could be measured by using only simple ultraviolet spectral photometer in regular experiments to replace complex and expensive instruments. Therefore, real-time F2 could be calculated by simple experiment for accurate modified ETR calculation in 1 cubic centimeter cube. So that is the basic measuring unit of chlorophyll, fluorescence meter, and the ultraviolet spectral photometer. So it can be easily calculated and measured. Well, from this formula, we can see the new F2 was changed with different cell density and pigment concentration, because optical properties, including scattering and transmittance of microalgal cells in suspension, will be changed in the whole growth period. And we can see the new F2 will be better to depict. Is just here. It can be better to depict the growth tendency of microalgae compared with the constant 0.84 and the absorption coefficient of single microalgal cell fixed on the film. However, what is the accurate relationship between modified ETR with new F2 and the microalgal growth rate? This research demonstrated that there existed a linear relationship between a non-modified ETR and microalgal growth rate in 1 cubic centimeter cube. In this relationship, the slope represented the converting ability of the light reaction intensity into a real biomass accumulation efficiency. And the slope is always greater than 0. So it represented the photosynthesis to drive microalgae growth. It must overcome the adverse effect from metabolic and respiration consumption. Light reaction must be stronger than the stress of those consumptions to make microalgal grow. Therefore, the slope and intercept together reflected the threshold value of the photoreaction, the dynamic potential required by microalgal growth. Experiments was carried out in different cultivation conditions with changing nutritions, light, and initial microalgal concentration, which showed that different initial microalgal concentration made no influence on this relationship, as well as light and nutrition only changed the slope and the intercept, but did not change the linear relationship. So adequate nutrition helped lower the threshold, while excessive light increased it. These above phenomena proved that the average state of nutrition and light exposure of microalgae caused all the microalgae cultured in the same state will share a same set of slopes and intercept, reflecting the linear relationship between photosynthesis and growth rate throughout the whole growth cycle. So the growth rate at a certain moment could be calculated by substituting the modified ETR in that moment into this formula. Based on those relationships and parameters in 1 cubic centimeter cube, this relationship could be generalized to any photoreactors with different shapes, sizes, and materials with different optic property. We made a software to calculate the actual absorption light of microalgae in every size photoreactors to calculate distribution of modified ETR and growth rate at each moment. Therefore, the average modified ETR and average growth rate of any shaped photoreactors at any microalgal cell density could be obtained to stimulate a growth curve for certain photosynthetic microalgal reaction systems. At last, by using the above calculation model, some difficult problems in microalgal research could be solved. Here we show an example to you. First, we have two geometrically similar column photoreactors. Because their ETR calculated by our software are totally different in this showed in this left figure. So at last, in the actual experiment, they have totally different growth curves, although with the same microalgal strains and almost the same experimental conditions. Compared with it, we proposed the concept of biological similarity. It can be explained as in two reaction systems, one with the same cell density, average modified ETR slope and interception were approximately equal, can be called biological similarity. At this time, we show an example on the right. The experimental phenomena for microalgal cultivation in those two reactors were similar as duplicates. No matter the shape of those two reactors were totally different. This concept solved the problem of experimental amplification that long puzzled researchers. As long as two reaction systems were biologically similar, then they must have a replicable experimental phenomena. Conversely, experimental phenomena in two systems that did not achieve biological similarity were incomparable. We can use this concept to scale up of our experiment from laboratory to the industry. If the microalgal industry with carbon fixation can be promoted a little because of this calculation model, that is our dream and honor. So thank you very much for your listening. That's all. Okay.
Thanks, Jenny, for your nice
Yes.
presentation. And I guess our audience may have a question for you. Any question for Jenny? Jenny, I just have a very basic question. So you developed a model for the photosynthesis for the micro growth. I mean, for the photobioreactor. My question is in your lab, you got such a setup, and then you validate your model by the experiment, or you just do the simulation compare with the literature?
I should say in our laboratory, we made some examples to show the actually, we just provide some concepts and we demonstrated the usage of those concepts through our experiments and our calculation. And we also try to try to make different experiments with even in the industrial scales to demonstrate it, and it also can be used to explain some the phenomena in other literatures, especially the totally different growth curves reported by other literatures. In the past, many researchers try to compare them and try to compare them, but our research showed that why those experimental results cannot be compared, because they have totally different cultivation systems. So because the optics are totally different, so even with the same microalgal strain, the experimental results cannot be compared. And the comparison between those kind of literatures are meaningless, maybe. So we just show this kind of concepts and ideas, and also we try to demonstrate it by different experiments, and both in the laboratory scale and in the industrial scale.